who to those joining us from United Kingdom, and good morning to those connecting from Mexico and other parts of the world. My name is Grecia Suarez, and I'm part of the team at the UNAM UK Center for Mexican Studies. We also joined by Dr. Antonio del Rio, Director of the Center for Mexican Studies, UNAM UK, who will supporting us moderating the Q&A session. On behalf of our center, it is a pleasure to welcome you today's seminar, part of our series Mexican Women in Science. This initiative seeks to highlight the important contributions of Mexican women in scientific and technological fields, and to create spaces for dialogue and exchange within the academic community. This seminar is also organized in the context of the International Women's Day, commemorated on 8 March, a moment to recognize the achievements of women, to reflect on the importance of advancing equality and visibility in science and academia. Today, we are delighted to welcome Dr. Ella Vazquez, researcher at the Institute of Ecology at the National Autonomous University of Mexico. Dr. Vazquez is a molecular ecologist whose research focuses on understanding how ecological and evolutionary processes shape the genetic patterns of wildlife populations. Her work spans areas such as population and landscape genomics, phylogeography, hybridisation, urban evolution, microbiomes, and zoonotic diseases. Together with her students and collaborators, she has conducted research across a wide range of natural environments, including tropical forests, islands, deserts, and freshwater ecosystems, working with different vertebrate species and focuses particularly on biodiversity conservation. Today, she will present the talk entitled Multiple and Diverse Impacts of Human Activities on the Genetic Patterns of Wildlife. Before we begin, just a couple of brief reminders for our audience. This seminar is being streamed live on YouTube, and we warmly invite you to share your questions in the chat through the session. We will have time for the Q&A discussion at the end of the talk. With that, it is my pleasure to introduce our speaker, Dr. Vazquez. Thank you very much for joining us today, and the floor is yours.
Thank you very much, Grecia. The pleasure is all mine. And good morning in Mexico, good afternoon in the UK to everyone in the room listening. And yes, as Grecia said, I'm work in the Institute of Ecology, which you can see a picture on the top, which is a really nice research institute. And the picture on the bottom is just to show you that we're set right behind or beside the botanical garden of the UNAM Botanical Garden, which is a beautiful, amazing place. And although I don't work with plants and I know very little about plants, going to the botanical garden for a walk is just a pleasure every day that we can do it. And also, as Grecia said, our lab is called Genetics, the Laboratory of Genetics and Ecology, because that's what we work with, all genetics and all about ecology. And I am passionate about animals. All of those pictures that you see in the screen are species that we have studied and that I have myself, most of them photographed except the frog. And we work in a very, very wide set of ecosystems and environments, islands, desert, forests, mountains, everything, everywhere, and doing all of that list of topics that we search and make questions and try to answer them through the study of the genetics of populations. And to put into context the talk, what I'm going to be talking to you today, I would like to start by mentioning that I imagine most of you, if not all, have heard what we are calling now the Anthropocene, which is called the Anthropocene epoch, which Anthropocene means the era of the human era, or the era of humans, and it's been put into the arena to highlight how humanity's activities, humanity's impacts have altered the entire Earth. It's reached all the realms and all the ecosystems on the land, on the sea, and it's just trying to make people aware of these human activities and how they have a lot of negative consequences on biodiversity. But in fact, we have been documenting. We really, long before talking about the Anthropocene, we already knew and have been documenting all people doing research in biology and conservation, that different human activities have had and continue to have negative consequences on all levels of biodiversity. One way of measuring that is how many species have become extinct. The International Union for Conservation of Nature, the IUCN, is the place where they have the most complete list of extinct species, also all the species that have one kind of conservation threat. And you can see I just put 3 different species, but they deer, sea lion, a rodent, a small mammal, species have gone extinct in all different taxa. And different studies have different measurements indicating that around 15 to 40% of species will go extinct by 2050. That is a horrible number, and it's not exact science, but it's very near that. And more importantly, it's not only thinking of the losing species, which is really, really worrisome, but there's also the extinction of populations and the loss of genetic diversity. So different levels of life organization that we are losing. And it's not only counting the number of species. We have to really focus on other levels. And again, different measurements, different numbers talk about a nearly 10% of genetic diversity that has already been lost across animal and plant species. And this is not only restricted to species that are threatened with extinction. We are losing populations and we are losing genetic diversity in species that are not even threatened. And another thing that is really important is that this continues. It's not, if you can see the times of the species have been declared extinct, you can see 2019, 2021, these 2 amphibian species were declared extinct, and that is a continuing process, sadly. And yes, different studies have documented this. I'm now going to focus more on the genetic diversity processes and how they can be diminished, genetic diversity and lost. We, a group of collaborators, we did an ample review of wildlife, natural wildlife, both plants and animals, mainly vertebrates. And we documented the genetic metrics they have been published and compared, and were able to determine that around 6% of genetic diversity in these almost 100% of 100 different species, 6% of loss of genetic diversity has happened since the Industrial Revolution. This was measured from the Industrial Revolution to around 2015, and that's the number we were able to calculate 6%, but it went to a staggering 28% if you think of, or if you refer to species living on islands, oceanic and continental islands, which is really, really, again, scary. So it is really important to grasp that losing genetic diversity compromises the evolutionary potential of these species that have their genetic diversity diminished, meaning that they're less capable of coping with, mainly with environmental changes, climate changes, that the greatest climate change ever we're living now. So they are not able to really cope because they have lost genetic diversity. So that is one direct consequence. So we really need to understand where the wild species stand in terms of their genetic background, how are they behaving in terms of genetics, in order for us to be able to predict how much they have maintained or are losing their evolutionary potential, and to how they are going to be able to stand on climate changes and environmental changes. And with all of this, with the main aim and objective of ensuring and trying to ensure the viability of their populations, if not the species as a whole, but at least of their populations. And that is what one of the lines of work we have in our lab. We are really, really keen on studying and determining the consequences of anthropogenic actions, anthropogenic activities, environmental changes on the genetics and the demography of wild species. And on the good side, or at least on the more brighter side of this talk, is that genetic information has been really key in terms of conservation applications. And I mean this because 20 years ago, biological conservation, biodiversity conservation didn't include genetics. They were more on the counting of number of individuals, number of populations. But now the genetic information is more and more incorporated into conservation programs. And genetic data indeed has contributed to understanding how these threats and what are their consequences on wildlife conservation. And they have also, these studies, these data are really motivating the perspectives of a potential solutions and for biodiversity conservation, and are guiding what we can do and how we should do it. And for that, in the following, I'm gonna be telling you about some of our work in my laboratory, focusing in these 4 anthropogenic activities, but the list of activities that are major causes of biodiversity loss and genetic loss, and that threatening our biodiversity is much more long than this one. But I will concentrate in giving you examples of how genetic information is obtained and what does it provide us in terms of what's happening to the species or the populations in nature, and how we can take that into consideration for conservation. I'm going to be showing you examples of habitat transformation, illegal trafficking, how invasive species also play a role, and of course, urbanization. And all the studies and examples that I'm going to be showing you have been done with many, many different students and colleagues. And I want to show my gratitude right here before I start to show you all the examples. So the transformation of habitat can be it comes in many flavors and many sizes and many forms. And it is really a strong negative influence, or has very strong negative consequences, especially for endemic species. Endemic meaning those species that are restricted to a certain geographical location, to a certain island, for example, or certain region within a country, that they happen only there. And if these are endemic species and on top they're isolated, well, the consequences are even worse. So what here is our first study showing you the Mexican country, the map of the Mexican country. Here we have the Caribbean. I hope you can see my pointer. And there is Cozumel Island. It is the biggest Mexican Caribbean island. It is big, but it's still an island where species are isolated. Cozumel Island has around almost 30 native endemic species, and one of which is the pygmy raccoon, Procyon pygmaeus, which we have been studying for many years, and it used to be distributed along almost all the Cozumel Island because it can survive in different habitats, deciduous forests, mangroves. It's very like cosmopolitan in terms of habitat. And we did our genetic magic there. We studied the many individuals throughout the island, and what we found, and I'm going to be showing you this kind of diagrams along the talk, is what this analysis tells us is how different these individuals are genetically. And what this graph is showing us is that we have the population of these species across Cozumel is actually being genetically differentiated. That doesn't mean there are different species. What it means is they are having little migration, little dispersal of individuals between these two genetic groups, and that has different consequences on the genetics of the individuals. And this separation is mainly associated to two main causes. One is here is the San Miguel Cozumel, which is the town on the island, which has been growing and transforming the habitat. This is the first or main cause that is separating these two genetic populations. And the other is that in the north, in this part, there is the pygmy raccoon has become touristic attraction, and they are being artificially fed and kept, and the consequence is individuals are not moving because they're having food. They don't need to go and look for food, and that has, on top of separating the distribution, has a very dramatic consequence, which is they're getting higher susceptibility to diseases, mainly because they get into contact with dogs. And so dogs are transmitting diseases to this part of the distribution. So you can see one habitat transformation and then other kind of activities have this genetic consequence. In another example, which also has to be with transformation of habitat and with an endemic species, but this is not on an island, not on a continental island, but some sort of island in the continent. This is the Cuatro Ciénegas Valley. It is located on the north of Mexico, which is a very desertic region. It is an amazing site. I hope you can see in the picture how we call it, like, seems like we are on the moon. It is really hallucinating. But what it is a set of different water bodies, water sinks, water holes that have been there for really long, thousands and thousands of years, and they are interconnected mainly through floodings. This is being a desert. It has a certain time with very high rainfall, short but very strong rainfall, and this gets flooded, and that's how it has been. Is the dynamic of this system. And here we studied the Kowalian box turtle, which is also an endemic species that is only distributed in this region. And again, the habitat transformation, the human activities that are impacting this environment is mainly water extraction. They are they have been for many, many years, extracting water for irrigating fields for cows and to produce food for cows and that sort of animals. I forgot the name. In the milk industry. So as one very, very sad example, we started working to study the Coahuilian box turtle in 2016, and this was one of the many water bodies we studied. And by the 2020s, it was completely dry, and all the population of the turtle that we had samples and we had been studying went extinct. So as I was saying, we can have extinction of populations also, and that happened in our case. And the genetic data, you can see this is a much more wider distribution compared to Cozumel Island, but we found exactly a similar pattern where we have two completely separated genetically populations, the yellow and the red one. And what this means is they are isolated between them. This blue arrow is this is the water body that went completely dry right here. So in the southern part of distribution of one of the genetic populations. This kind of diagram, what it represents is how historically and also contemporary these populations were connected, how much dispersal of individuals there is, historical and contemporary. And actually what it's telling us is that only the two nearby populations, water bodies, are at present having this connectivity. And that means there's very, very significant genetic diversity loss. There is not genetic connectivity. Populations are getting isolated, and that has a lot of different consequences. And all wildlife, all animal species are key to the ecosystems where they live. Some have specific roles that they play. And for example, Terrapene coahuila, this turtle, is a barometer of the health of the ecosystem. If we find turtle, it means the water is pristine, it is in good condition, and if it is absent, it's telling us something is wrong with the ecosystem. So you can see a lot of problems here as a consequence of subtracting water and desiccating water bodies. And now Terrapene coahuila is extremely endangered with extinction. Habitat transformation, we can study it in all different scales. And now I'm showing you an example of a species that at least had historically a very, very wide and connected distribution. This is the mantle howler monkey. We have 3 non-human primates in Mexico, and this is one of the 3 species. It's a big animal that is strictly arborical, meaning it lives only on trees, so it needs forested habitats in a very well, at least a good conservation status. And despite historically it ranged all over this area, now we have isolated, fragmented populations, as you can see in the different points. And our genetic data is corroborating their isolation. This is the Santa Marta Mountains in the northern distribution of this species. And this is one genetic group that is clearly separated from the rest. And the consequence, why we have this fragmentation and isolation is because for more than 800 years, this has been transformed mainly for agriculture, but also for cattle. And the habitat has been highly deforested, and meaning this species has lost the habitat where it can live. And we are seeing now different consequences. This is a very social species, like almost all primates, and this separation is disrupting their social behavior, which is having very, very bad consequences. Is also having higher disease susceptibility, because it's living in very small fragments and it's not getting enough nutrition. And so it's also a species that is really endangered. And it's important to mention, it is a very good seed disperser. It is it's crucial for the forest environments, the presence of this species and the consequences of not having this species. And it is reaching, as usually happens, even us, because there is less forest and people depend on forest for humans depend on some of these forests for food and for wood. So losing all these species, and the functions it has in the ecosystem has negative consequences all the way up to us. Okay, I'm changing now to a second of my examples, which has to do with the intentional or unintentional movement of individuals, also trafficking of wild species. And in this special case, I want to tell you about hybridization. Hybridization means the two different species come into contact where they not usually should, and they can, in this case, they can reproduce and have fertile offspring. And so we have hybrid individuals with many different consequences. But let me just tell you very quickly the story. We have two crocodile species that distribute in Mexico. One is the Morelettis crocodile, which is its natural distribution is the Gulf of Mexico and the Yucatan Peninsula. It is considered endemic species, a regional species, because it's mostly distributed in Mexico. And then we have the American crocodile, which is distributed in the Pacific, and it has a very, very wide distribution. But at least these two species are separated in the Pacific. Morelettis does not live naturally in the Pacific, and that is important to keep in mind. We have been studying the crocodiles for many, many years, so we have data all across the distribution of both species, but I want to show you here one that pertains to intentional movement of individuals. So this is a national park, El Cañón del Sumidero, that is on the Pacific coast of Mexico. And we have data from there, which we expect to have only American crocodile because that's the natural distribution, which is effectively the case. And we have data from two artificial farms where they grow and maintain these species because it is used for for meat and other aspects. And we did the genetics, and what I'm showing you here is that in the national park, we effectively have only the American crocodile, the species you expect to have there, which is in red. But in the northern farm, we have most or all individuals, at least the ones that we were able to sample, they are from the American crocodile, which shouldn't be there. But you can think, well, it's the American crocodile, they have it in farm, they are keeping it safe there. But the problem was in Villaflores, the other farm where we found that they have American crocodile, but it's obvious that they introduce both species in the same place, and they have been hybridizing, and we have hybrids now inhabiting, and that has many different consequences in terms of genetics and in terms of maintaining this species. And it is not just there. It is something that's happening more often than we wish. This is the area that I just showed you, but there are at least two other sites that we've found hybrids, meaning there is some of the Morelettis have been moved to these areas because it doesn't naturally occur. So we're having hybridization as a consequence of of animal farming and a human activity, and animal farming mainly for meat and for poultry. So, and this, again, not time to tell you all the extreme negative consequences it can have on the natural distribution and natural survival of this species. Illegal trafficking also has to do with the pet trade. And the pet trade is one of the worst human activities and human effects, especially in terms of invasive species. Invasive species are species that are transferred from their natural distribution to a place or places where they do not naturally occur. And this happens from species from North America, from America to Europe, from Europe to Asia, from Africa to that level of movement of individuals. And the case study I'm showing you here is the red ear slider turtle, or the Japanese turtle, they used to call it, very, very popular to have as a pet during the 1970s to probably early 2000s. It was sold in all of the pet stores in the world, and it is now classified as one of the 100 worst invasive species in the world. It is really tragic because invasive species displace native species where they arrive, because they are more successful, because they are highly competitive, for many reasons, and that's why it's a problem to have invasive species. The Trachemys scripta elegans, the Japanese or red-eared slider, its not native distribution is in the eastern of North America, right down to the border of Mexico. That is where it naturally occurs. In Mexico, we have 3 other species that are congeneric, meaning is Trachemys something else, that naturally occur in this geographic gradient from northern Gulf of Mexico all the way through, again, to the Yucatan Peninsula. And we have another endemic, it's called Trachemys taylori in Cuatro Ciénegas, another endemic turtle in the valley that I was showing you just now. We also have done genetics throughout all the distribution of these 4 species. And one first thing that was interesting to find is that the individuals from the native distribution of these species are genetically absolutely different from the same species that is distributed as an exotic species in Mexico. We had some samples, and one possibility was that they weren't different because they could be even natural migrated. They don't migrate. They're intentionally moved, and we know that this species in Mexico, it is genetically different, so it is classified as an exotic invasive species. And we also did a lot of genetic analysis with the 4 species that I show you in this gradient. This is again the Japanese, the one that is worldwide invasive, and these are the ones distributed in Mexico. And these graphics, what they are showing is they're based on genetic information, and they're simply put, they're saying that the species, the natives in Mexico, are absolutely genetically different, and there's no gene flow between them. When we compare the Japanese turtle with Cataspila, which is the one distributed right here in the Gulf of Mexico, and the same happens if we compare it to the more southern turtle species. So they're very, very different. And this is interesting because it sort of functions like a filter. The Japanese turtle hasn't been successful in naturally invading Mexico, because it has these 2 other like sister species competing and defending their distribution. So it's a good thing for Mexico. But see what happens. But it has been, the Japanese turtle is one of the most successful invasive species in all the world except Mexico. And here, what I'm showing you is not based on genetic diversity, but it's based on the ecological and environmental variables that define the environment where these turtles can where these turtles survive and reproduce and are successful. And what I just want you to catch is in red, we have this combination of environmental variables that define the niche of the Japanese turtle, and in the other colors are the ones in Mexico. And you can see that they overlap, but not much, meaning their ecological niches are different. So also contributing to the Japanese turtle not being able to invade. But see what happens in the other areas where it is invaded. It has invaded. And we compare these ecological niches with endemic native species in these different places, Europe, Asia, Australia. And you can see how in red, again, the Japanese or the red slider turtle, and in most of them, the niches are almost 100% or very much overlapped, meaning that's the conditions where the Japanese turtle has invaded are good for it. So genetically and ecologically, it is very successful, and that's the scenario for most invasive species that are very successful where they are distributed. And for the last theme, I told you I was going to talk a little bit about urbanization. Your urbanization is, for me, is the most extreme of the gradient of human habitat transformation, because we're going, as this very neat diagram shows, we go from what it is a natural environment with the fauna and the flora that belongs there, slowly or rapidly, it depends where we are, modifying, subtracting, deforesting that environment, that habitat, until the end of the gradient, where we come to these big cities with impervious surfaces where nothing can grow, everything is it's pavement and buildings. And some species can adapt, that are in the wild, can completely adapt to these urbanized environments. Some others can more or less adapt. For example, this bird, because it is able to fly from more natural places to the city and go back. And there are other species that absolutely cannot. And we have, and importantly, urbanization is also one of the factors that are accelerating in the highest grade the changes in genetics and in evolution in plants and animals. It is such a strong force that either the species just don't survive and disappear, or some adapt really fast. So in terms of evolutionary studies, it is really interesting setting, but still it's a very, very bad setting for many species. And we have this, I'm showing you very briefly, we studied this lizard, the mesquite lizard, that distributes in the mountains of the middle of the country. It's called the Trans-Mexican Volcanic Belt. All along this, it's very wide distributed. It's a generous species, which you would think it doesn't have much an effect on it. And here we didn't compare to urbanized areas, just to agricultural lands, comparing to forested lands. And what we found is genetically it is getting different, is losing genetic diversity, but also, and very interesting, they have changed in size. They are bigger and more chubbier in the areas where in the agricultural areas, they don't have this protection of the forest. They have to thermoregulate differently. So we are seeing consequences not only in the genetic level, but also in the morphological level. And we're going back to finish. We're going back to Cozumel Island, where we are here combining two things: urbanization, but also invasive species, and in these isolated environments like islands. Two of the most successful and widespread invasive species worldwide are the black rat, Rattus rattus, the Norwegian rat, that is Rattus norvegicus, and the house mouse, Mus musculus. These two species are they're originally from Asia, and they have invaded all over the world. And they reach, not, of course, they also reach Cozumel Island, and we studied their distribution and their genetics. And despite their how generalist they are and how they can survive almost anywhere, I'm sure all of you have seen at least a rat on the cities. We found genetic changes and genetic differentiation at the level of, for Mus musculus, at the level of the town, the San Miguel Cozumel that I had mentioned. It is very confined, associated to the urban landscape, but it has genetic differentiation. And Rattus, which is able to disperse, is bigger, and it is able to disperse much more. It's distributed in a wider range, but it also has differentiated. And I want to explain you a little bit more of the differentiation of Mus musculus, the one that is very small and is confined to the entire urbanization region of the island. And what is really interesting, at least we were very happy with the genetic data that we obtained, was that there are three genetic groups. We call them San Miguel 1, 2, and 3. And you can see the colors. And we were able to match these genetic groups to how the transformation of the island has happened from before the 1970s. San Miguel was a very small town, and they have a landfill, an open landfill. All the garbage was transported to one place that was sky open. And this is the first oldest genetic group that we can identify. And then between the 70s and 2000s, there was an increase in tourism. The city started to grow. And then we have this second genetic group that is genetically differentiated. And the last one is, the blue one is the more recent genetic group that coincides again with a new growth of the island. It was influenced because the tourism started to be really massive. We're having these big ship cruises that transport not only people but also rodents, and a growth in infrastructure. And so these genetic groups, despite this species is very tiny and is only distributed in the city, it is actually genetically differentiated. So there's also other kind of things that you can describe from your genetic data, and this is a description of the invasion history and evolution of this species. For the very last, I don't know if you're familiar or if you've heard of the One Health approach, which is very recently established, and it means that in order for us to have this harmonic relationship with the earth and with all the species, humans, and all the species that live on it, we need to have good human health, good animal health, and good environmental health. These three need to be entangled. They are not independent, and if we are missing one, we're missing the three. And one focus on studying this is studying the microbiome, which is the community of bacteria, archaea, virus, fungi that we all have in all of our bodies, in the gut, in the mouth, in all of and not only us, but all organisms, plants and animals have microbiome. And we're starting to study wildlife microbiome. And in this very brief example, I'm not gonna describe that much, but here I come back to our primate species. The one I show you the study, the mantle howler monkey, but we also have studied the black howler monkey and the spider monkey, our 3 species of Mexican primates, which, by the way, these two hybridize naturally, and so we have data for the hybrids. But what I want to focus here on or highlight is how the microbiome is very different among the 3 species. And here we can identify, and it will be too long to describe you how, but we can identify individuals, these are, every bar is one individual, that have altered their microbiome, either because they have over-representation of one genus or one species of bacteria. Here again, this individual is very, very diminished the diversity of its microbiome, only two species or 2 genera. So this is another approximation that we can approach to see at the level of wild species and at the level of their microbiome, in this case, the gut microbiota, how healthy they are, how deprived they are in their gut diversity, which has direct association with the health of the individual and the health of the ecosystem or the habitat or the region where they are. And we are also getting into very recently into these kind of studies, and there's also a lot of means to help or show what is happening at the level of individual, and we associate this also with genetics and finally with proposals of conservation. And with that, I will thank all of these institutions that have contributed and helped us to be able to do all of these studies and be so happy in the field and in the lab. And thank you all for listening.
Thank you very much, Dr. Vazquez. We have some questions here in our networks. One is, how quickly can wildlife populations show genetic responses to the environmental change caused by human activity?
Yeah, that is an excellent question. And the answer is going to, as always, be it depends. It can take a long time, especially in where individuals or species have long life expectancies, where they have slow reproduction rate, where they have a low growth rate. For example, in the primates, they compare, for example, with rodents. Rodents, they live 2-3 years. They reproduce before they reach 1 year. They have more than one reproduction event per year. Those demographic characteristics are allow for genetic changes to reach very quickly. In comparison, if we talk about the primate species, or for example the jaguar that is on the drawing there, the genetic changes take much longer to be that we are able to document. Although it also depends on how strong the effect or the force of change is. So maintaining these differences, because due to the life history of the species in question, if the force is really strong, and we will be able to see these changes more quickly if in comparison with where the effect is not as strong. So it's like considering those 2 factors.
Okay, thank you. Here we have another question. How can genetic data help identify population connectivity or fragmentation in altered landscape?
Well, that's the beauty of genetic data completely. What you need to do is to have genetic data of the different individuals or the different populations that you want to know how well connected they are, or if they are not, because we measure gene flow in a direct or indirect way, we estimate gene flow. And as the diagram that I was showing for the box turtle, that kind of analysis allows us to be very certain and very precise, if you want, to measure these populations have been connected and are still connected, and there's this average of gene flow between them and among them. And yes, we there's even a term called functional connectivity, which means you know the genetics of the connectivity, because you can have, for example, if you mark individuals, so you use radio collars or that sort of technology, you can actually measure how one individual moves, but that is structural connectivity, but we measure the functional connectivity. And the beauty of that also is because we can, depending on the genetic marker that you use, you can differentiate between historical connectivity, meaning the historical of a movement of the populations that you're studying, and also the very contemporary current movement. And usually there is a disconnection of that, like we show with the howler, the mantle howler, that I didn't show that data, but we have data that represents how historically connected they were in terms of genetics and how they are not in contemporary terms.
Okay, can you explain for a Legos like us how is possible to define the age of a population in terms, for example, the Cozumel rats? Why you say this population is ancient than this older, this one, and well, for me, it's not easy to
Of course, no, yeah.
distinguish.
In the case of the house mice in Cozumel, we didn't do what I'm going to tell you how we do it, and then I'll come back to Cozumel mice. We usually use genetic markers that have very low evolutionary or divergence rates, and we build phylogenetic trees. And we are able to, using the rate of the diversification of the gene or genes that we are analyzing, and in combination with, for example, fossil dating, so we can know a fossil from here. In the case, for example, with the box turtle from the Cuatro Ciénegas, Coahuila, we did this kind of analysis, and we were able to date with certain error margin, of course, that that population established around 10 million years ago in the valley, and how it has been, how it was evolving, and how it came to be the species that is now endemic and restricted to the valley. So that's one way of doing it. In the case of mice, we didn't actually have the dating per se, but knowing where these genetic clusters were very well defined. For example, the very first one, we know that the possibility of these animals to reach the island need to be through commercial shippings and that sort of transportation. So that's why we say before the 1970s, this was established. When, we don't really know. But the interesting thing was how we were able to match how the second group was an area that didn't exist. It wasn't urbanized, wasn't transformed or until the 2000s. And that is another genetic group. So this was more of a process of matching genetic groups with what has happened on the island. We didn't actually measure the timing.
Okay.
It's more of association.
Here we have another question. Can you discuss how human actions could increase genetic diversity?
Yes, sure. And it's one, that's a nice question, is one of the means that conservation genetics and conservation of biodiversity are actually trying to safeguard some species. If the important thing is you need to know the genetic information of whatever you're trying to increase genetic diversity. But well, you have a population of a number of populations of a species they are declining, they are losing genetic diversity. You can translocate individuals from another population to these areas in order to make them reproduce and increase their genetic diversity. But this has to be done really carefully, because if you mix individuals that are genetically, it's called outbreeding depression, that if you try to mix genetic compositions that do not match, despite they are the same species, you can have the contrary consequence, diminishing the genetic diversity. But yeah, it is really possible. You just need to know more or less the genetic pedigree of your individuals, and that's what, for example, zoos try to do. They reproduce in captivity, and they reintroduce individuals to the populations they are trying to increase genetic diversity, and is one of the best means of maintaining genetic diversity or increasing. Just need to be done very carefully.
Okay, thank you. And I am sure in the cities in Mexico also, for example, in Cuernavaca, we have some squirrels and another animal as zarigüeyas or and others. And they, I am sure they also have this effect of islands, but inside
Of
a
course,
city.
yes.
Yeah, there are studies about that.
Yes, yeah. It is called urban ecology and evolution, which is really I'm really passionate about it, and with these mixed feelings, because they are evolutionary laboratories. It's incredible what you see, how quickly evolution and local adaptation can happen. And yes, well, there are many studies, and to tell you one in Mexico City, and yeah, especially in the southern part of the city, but all across, we are seeing an increase in the presence of cacomixle, which is this kind of a lemur with glass, yeah, beautiful mammal, and because the natural distribution, it's been fragmented, the forest is being deforested, they are actually moving towards more along the city, and now we are seeing them in our gardens, and there's not in my, it's not me, but there's a group studying how they are genetically and ecologically, and now we have genetic groups, one in the north, one in the south, one in the southern of the city. They are yeah, moving in these, as you say, islands within the city, and they now are showing these genetic differences. Yeah, but yeah, there are many examples of with reptiles, birds, plants.
That's okay. And I am sure the infrastructure of the cities has an effect in the wildlife. But what's about our pets, cats and dogs? Can you say something about maybe cats,
But did
maybe
say
dogs?
what about what their genetics? I don't know what.
No, about the genetics of our pets, but the
Uh-huh.
threats that
Yes.
our pets do to in the wildlife.
Yeah. Yes,
If
of
we can
course.
say wildlife like squares of cacomixles or zarigüeyas.
Yeah. Well, yeah. I know a lot of people love cats and dogs, and they don't like to hear much of it, but they are very bad. And you know what? Well, cats in particular, they if they are left free to go everywhere they want, they have this, well, if not instinct, at least, this behavior of hunting, and they hunt small birds, they hunt lizards, they can hunt even small mammals, and so there is and it's measured and it's shown in scientific literature how they have a very, very big negative impact, diminishing native populations of wild fauna. So that would be probably the most, the strongest or most important impact of domestic cats. And in terms of dogs, they transmit a lot of diseases, like the ones I was saying in Cozumel, how they have transmitting rabies, and there's another very common disease in dogs that they transmit to just by biting. In this case, the raccoons, and they also, when they become feral, when they have been on the wild long enough, they become hunters also. So there will be diseases and hunting.
Okay.
So we have to be very careful to not let your cats roam free, and don't leave your dogs don't abandon your dogs. No.
Okay.
Mhm.
Well, Ella, thank you very much to quit with us. Really, we appreciate this wonderful conversation about what we are doing with the wildlife in many places, not only in the wild, but also in the cities. Thank you very much, and we appreciate that you are with us. And let's say this is a seminar that formed part of our Mexican Women in Science series, organized by the UNAM, United Kingdom, and held in the context of the International Women's Day of the HMRs, which encourage us to recognize and amplify the contribution of women in science and academia. We invite you to continue following our activities and coming seminars through our websites and social media. Thank you again for all that is attending. Thank you, Ella, for this wonderful presentation.
It was my
Bye.
pleasure. Bye bye.